Well, thank you. I might just request assistance in bringing up my PowerPoint. And in the meantime, I'd like to thank very much Patrick and Katie, the organizers, Master at Massey College, Miguel, or is it my angel? Oh, okay. You are my angel. Thank you. And also General Valera for the introduction this morning. I'm going to take a step back and to broaden in some ways the discussion, which is focused very much on exposure to extreme violence and images of extreme violence and PTSD, anxiety, depressive disorders.
Sorry, I don't find it here.
Then I would have a slide right there. But these, stepping back from the journalists and from the soldiers, there are many people in our society who do develop mental illnesses, such as anxiety disorders, depressive disorders, even PTSD, without having been in the journalism newsroom, without having been in Rwanda or at war in another country. Thank you. I just give you the percentages here. Diagnosed. Yes. Diagnosed during with a diagnosable mental illness during any given year. It doesn't necessarily mean these people are receiving treatment, but just that they would have the symptoms that diagnose them. Is everyone good for the audio? I see. Okay. No. Yeah, up? Okay. So the question is, are we all equally vulnerable? So this picture I chose, the Edvard Munch. He painted The Scream, and he spoke of his own experience. My father was temperamentally nervous. From him I inherited the seeds of madness. The angels of fear, sorrow, and death stood by my side since the day I was born. Well, part of my research, so I'm a neuroscientist. I do neurophysiology, and I'm going to talk to you today about work that looks at development and how that may increase someone's vulnerability to acquiring psychiatric illness later in life. I'm very interested in what is the brain function like in a vulnerable individual. And the reason why this is so interesting to me and the people in my lab is we hope that by looking at this neurophysiology, we may get clues to develop better treatments. So where do we look in the brain for this vulnerability? Well, the prefrontal cortex, as you see right there in the screen, is the area that is particularly interesting. It's the part of our brain up here. I'm used to having a laser pointer, but I'm going to just make do with my hands. And now I've been told that I need to be, thank you. I'm also used to pacing. So I will stay by the microphones, but hopefully this will do my pacing for me. So the prefrontal cortex, this part right up here, is how we interpret the world around us. It is how we assess risk and reward, which is something that's very important to do in war zones, but it's also very important to do in everyday life. It's how we concentrate or pay attention. It is necessary for us to inhibit impulsive behavior, thinking of that big glass of alcohol that Dr. Feinstein showed. It has also been shown recently to be increasingly important for memory retrieval and memory generalization, selecting the most relevant memories to bring to mind and preventing intrusive memories from coming into one's experience. And the prefrontal cortex manifests these effects by controlling other brain regions. So I just give you a sampling here for those of you who are not neuroscientists, so many words, but what I can tell you about some of these regions are what they mean to everyday life. So the LC is involved with norepinephrine, so your fight or flight response. The Raphe with serotonin, which many people think of as a very benign, happy neurotransmitter, but I'm going to show you perhaps the dark side of serotonin today. The VTA, dopamine and reward, and also aversive memories, the amygdala, our center of fear, and the thalamus, which is what is the center through which all of our sensory information goes through on our way to the cortex. The prefrontal cortex communicates with all of these regions, and they communicate back. So with the brain, it's very difficult because there's all these bidirectional communications. And so anyone could ask a neuroscientist, well, why do you start here? Why don't you start here? Well, you have to start somewhere. And so there's been a lot of research over the past 20, 30, 40 years in psychiatry to show that there's abnormal activity in the prefrontal cortex in psychiatric disorders. Now, there's a lot of controversy about this, and some people have their pet regions and their pet hypotheses about whether it's hyperactivity or hypoactivity or hyperactivity here and here and hypoactivity there and there. I won't get into those distinctions right now. What I will get into is the question of what comes first, abnormal brain activity in the prefrontal cortex or the mental illness. It's hard to do with humans. I mean, I'm a neurophysiologist who works with rodent models, or preclinical models, as Dr. Zan would say. But with human studies, even human imaging studies, like I used to do at CAMH when I was a student here at Massey College, it's very tricky to do a before and after. As Dr. Feinstein said, of all of those journalists who are being exposed to those images, only a small subset of them are developing intrusive PTSD. Many of them are resilient to it. Now, there was a couple of interesting studies recently by scientists from New York, and they looked at people, soldiers in this case, who went to Vietnam. And in a sense, what was very cool about this study is that they got to get a glimpse before. Now, how do you do that? You know, it's brain imaging before a whole bunch of soldiers go off to conflict. Well, no, they had a twist. So they found soldiers, some who were in combat in Vietnam, who developed PTSD, and some who were in similar levels of combat in Vietnam who did not develop PTSD. And the one thing that these two groups had in common was all of these gentlemen had twin brothers back in the States, identical twin brothers, who had not been exposed to conflict. So that's the very clever way of getting the before and after. So when they did brain scans of these twins, they found that in the soldiers who went to Vietnam, the ones that had PTSD had abnormal prefrontal brain activity. In this case, it was elevated within a certain region. Whereas the soldiers who went to Vietnam and did not get PTSD had much lower, significantly lower activation in this region. So the question is, what happened with the twins back home? Was this hyperactivity in the prefrontal cortex a symptom of the illness, or was it a predisposing factor? Well, it turns out that the twins back home, who were the twin of the brother who got PTSD, had very similar levels of heightened brain activity in this region. They then, in a later publication, this is Shin et al. 2011 and Shin et al. 2009, they went on to look at activation in a functional MRI study, and they were looking at emotional memories intruding on a cognitive task. And again, they found a similar pattern, where there was a vulnerable pattern before the person went off to war and before they developed PTSD. It's a small study, but I found it very compelling. And it's something that is very inspiring in terms of the work that I'd like to present to you today. When I was asked to present, I knew it was going to be a mixed group. Dr. Zan said, there'll be people from many different backgrounds. And I thought, oh, wonderful, I can give a nice general talk. And then she's like, and there'll be some expert scientists and the dean of the School of Medicine. And I thought, oh, okay. Well, so I wanted to talk about some work we did that I can give you a glimpse of what we do, but also what some of our collaborators have done to make it more general, to understand the bigger picture. And I say from the beginning, and I'm talking about development, this is not only the beginning, but this is the beginning of research into this area, trying to understand what is the neurophysiology of vulnerability. There is much, much still to be done. So the idea, one of the key people in starting this area was Michael Meany at McGill University, who described the idea that there could be a vulnerable period, and he started doing animal studies to look at this, where something happens early in development that makes you more vulnerable to stress later in adulthood. And so he was the one who started doing some experiments with early life stress, and it's been done by many groups since. The idea is that the human or the animal might undergo a critical period where something is happening in the brain which later on may render them more vulnerable to developing psychiatric illness. So what to look at first? We talk about looking at the prefrontal cortex, check, but what to look at in the prefrontal cortex? Well, prefrontal serotonin levels increase with stress, and especially the inescapable kind. I think I was so taken with this experiment, it's from Bland et al. 2003. See, I went from Mac to PC, and I think there may be a few glitches. But here you show a rat without any stress here at the bottom. This is looking at the serotonin levels in their prefrontal cortex. In black dots, you see the rat exposed to inescapable stress, one that it can deal with and get away from. And you see its serotonin levels go up, and this is over minutes. Whereas on the top, with the open boxes, you see the rat serotonin levels when it was faced with an inescapable stress. So serotonin is not just a benign neurotransmitter. Many people think it's happiness, and I should take my tryptophan supplements to help out. But it's also a neurotransmitter, neuromodulator that helps us cope with stress. So serotonin receptors are targeted by the most effective treatments for several major psychiatric disorders. And you've all heard of Prozac or SSRIs. The effects are very well characterized in the RAFE, however, the effects of these medications and also serotonin itself on the prefrontal cortex are only beginning to be understood. So within the prefrontal cortex, we have two major types of serotonin receptors: the excitatory 5-HT2A receptors, which I'm going to talk about the most today, the inhibitory 5-HT1A receptors, which we do a lot of work on and have published on recently, but that work is more technical, and I would like to keep this more general for today. So what are these receptors like in the stress-vulnerable brain? So we did whole cell recordings in a rat brain slice, and we had many animals from control litters and many animals from early stress litters. So they were all housed at the same time. I won't go into the details of this, but this is just a separation from the mum, 3 hours a day for a period of time. And it renders the animals much more vulnerable to stress and developing symptomology or endophenotypes of depressive and anxiety behaviors, because of course we don't know if a rat or a mouse is depressed. To give you an idea of what the typical serotonin response in one of these neurons would look like, it's an outward current. It's actually mediated by one of those 5-HT1A receptors that we were talking about. And when we started this work, this is what we were expecting to see. So with the controls, we recorded, it's all these upward directions. So this is a potassium current that helps cool down neurons and bring them back to resting potential. In the early life stress group, so these are rats that had been exposed to stress when they were young, but they are now being recorded as adults. They had quite a different picture. So they had these 5-HT1A responses up here, and I'm not going to show you a lot of the pharmacology. You'll have to take me on faith on that. And this, these responses down here, which we hypothesized were 5-HT2A responses, these are inward currents or excitatory currents, currents that if the neuron is allowed to go and fire action potentials, these ones would be driving them towards action potentials. So what happens if you selectively excite just those receptors? You can do that with basically a psychedelic hallucinogen. So DOI is like LSD, it's not as strong, but it stimulates just the 5-HT2A receptors. And you can see here it has a long slow inward current. And this effect was much, much greater in the animals that had been exposed to early life stress. So we have animals who are both adults, but they only differ in the fact that they had a stress treatment when they were young. And their 5-HT2A receptors seem much more powerful or much more able to exert electrophysiological effects on their prefrontal cortexes. So what happens when you look at baseline activity and the effects of 5-HT2A stimulation in vivo in these rats? So if you just look at baseline activity as assessed by immediate early genes, or ARC, you can see that the controls in the early life stress, they're not that different. There's a little bit higher in the early life stress, which is a little bit similar to some of the imaging work that's been done. If you treat controls with DOI, so the psychedelic hallucinogen that stimulates 5-HT2A receptors, you increase activity in the prefrontal cortex. But in the early life stress group, you really increase activity in the prefrontal cortex. So it's not just our neurons that we're recording from. These are the animals that have had their 5-HT2A receptors stimulated when they're alive. Now, are there changes in gene expression in the prefrontal cortex in these early stressed rats? So broadening this out still again, so we've been talking about how the 5-HT2A receptors seem to exert more powerful effects. What else is going on? We have some really wonderful collaborators at the Tata Institute in India who did a microarray on the cortex of these rats, on prefrontal cortex, and they found many, many changes in gene expression. One of the things that really caught my eye was this one on cellular developmental processes. And the reason why but there's also genes that are different in terms of response to stress, response to external stimuli, a lot of the neuromodulation genes. But the development really caught my eye because 5-HT2A receptors are very important for exciting the prefrontal cortex in development. So until postnatal day 15, which is very young for these rats, the majority of the neurons that were interested in prefrontal cortex are wildly excited by serotonin through 5-HT2A receptors. So it's as if in the healthy rats, this has gone away. This was something that happened at some time in development. Whereas in the early life stress rats, in the early stressed rats, this developmental manifestation has kept going. We wondered if that might be true for other things as well. So there's some validated genes in this group that were upregulated by early life stress, and they're typically only expressed early in postnatal development, but they continue to be expressed in these rats that are vulnerable to stress later on. And we wondered if you could mimic these changes by stimulating 5-HT2A receptors. So this is the microarray, and lots and lots of bars, eh? So the yellow here of the control is that there's not a lot of genes that are unusual because this is the control group. By contrast, these are genes, so each line is a different gene. And if they're red, they're up. If they're green, they're down. And this is the early life stress group. And so comparing each gene to its similar gene within the control group, you can see that there's a very, very different picture. So there's hundreds and hundreds of genes that are different if the animal was exposed to early life stress. Interestingly, if you take a control animal and you expose it to the 5-HT2A agonist, or the DOI, you can give a very similar, somewhat similar picture. And that's because the 5-HT2A stimulation is giving you is turning on some of the same genes. Which raised the question in our mind, can we restore normal gene expression by blocking the 5-HT2A receptors? And this is work done by our collaborators. And they found they could. So if they treated with a 5-HT2A antagonist for 6 days, many of the genes could have their changes reversed or normalized. And I just give you a few ones of the validated genes. So the 4 conclusions about the vulnerable prefrontal cortex: that there's abnormal serotonin-elicited excitation in adulthood. And as we talked about, serotonin is something that gets driven up by stress. So the response to stress is likely different in these animals. A large number of genes are involved in cellular development and excitability, and these ones are upregulated. So there's something fundamentally different about the vulnerable brain. And some of these changes can be mimicked by stimulating 5-HT2A receptors, and some of the changes can be reversed by blocking 5-HT2A receptors. And that raised the question in my mind, would these mitigate the vulnerable state? Looking into this in terms of human work, there's been a little bit of work that's been done with medicines that might block 5-HT2A receptors. There's a suggestion that this has an effect, a positive effect, in people who have PTSD who are resistant to current treatments. But I think it's a field that would be interesting to follow up with more study. But I urge caution, because the 5-HT2A receptors are very, very important. And having stronger 5-HT2A receptors might not just make you more vulnerable to devastating consequences of seeing violent images or being in violent situations or even everyday life and stress, but these receptors may be critical in making some of these people the excellent journalists that they are. And the excellent people they are, and the excellent scientists, and the excellent soldiers. Because these 5-HT2A receptors are involved in anxiety and risk assessment. They're involved in attention, they're involved in cognitive flexibility, and recent work by a scientist in Buenos Aires suggested that they're also involved in memory retrieval, selection of relevant memories to retrieve in particular. So it suggests that these people who are more vulnerable, there may also be some very positive things associated with the vulnerability of their prefrontal cortex. And that's why I urge there to be future research, and my lab is very keen to continue our work and to look at the interaction between early life stress and adult stress to see what happens in terms of the neurophysiology of the brain. But most importantly, what we'd like to understand is, are there treatments that can be used that are not such sledgehammer treatments, treatments that are able to target something that's really wrong, as opposed to a receptor that may be what was helping to make the person such an incredible person before they develop the devastation of a psychiatric illness. Oops. So I'd like to thank the people involved in this particular study. I have a wonderful student, Natalie Goodfellow, who did the majority of the work we talked to in collaboration with scientists at the Tata Institute in Mumbai. And Madhu Bankareddy and Vidita Vaidya. Our funding agencies, CHR and NSERC, and of course my whole lab for providing a rich and fertile environment to pursue this work. Thank you.